So in this next video, we're going to be talking about the endomembrane system. So let's talk first about proteins and how they're made and how they can be transported into organelles. So the default location for protein synthesis to occur is in the cytosol. But this poses a problem for proteins that need to cross over a membrane or embed into a membrane, because these proteins on the outside are largely hydrophilic, and it would not be favorable to pass through a lipid bilayer. So the cell has a solution to this problem, and that is that the cell can make the protein inside the ER, or they can keep the proteins unfolded until it can be passed into the organelle. And this is what happens in the peroxisome and mitochondria and chloroplast, this last one here, but we're not going to talk about those today. The mitochondria and the chloroplast can also make their own protein because they have their own genome. But what we're going to focus on are the proteins that are made inside the ER. And the fate of these proteins is to end up in the ER, the Golgi, lysosomes, or the plasma membrane, or secreted outside of the plasma membrane as well. So let's talk about the experimental evidence that led us to really understand this pathway that's the endomembrane system. And so scientists did this experiment called a pulse chase experiment. And what they do in this experiment is they take this culture of cells here and they add in essential amino acids, so that all the proteins that are being made in the cell are taking up these amino acids and synthesizing proteins. After a period of time, they do what's called a pulse. And in this part of the experiment, they add radioactive amino acids. And so any proteins that are made during this time are radioactive. And so we can follow them using a variety of techniques like microscopy. Then, in this period called the chase, they add an excess of non-radio labeled amino acids, so that now all the proteins that are being made are no longer radio labeled. And so what this allows us to do is follow a group of proteins that are made in a given period of time. And when scientists did this, they found out that some proteins move from the ER to the Golgi to the plasma membrane. So let's take a closer look at what we know now to be the endomembrane system. So proteins move from the ER to the Golgi. That's the first step. And some of these proteins can be secreted outside of the cell. Other proteins, especially those that are amphipathic, end up getting embedded into the plasma membrane. A third fate for proteins made in the Golgi is trafficking to the lysosome, and we'll talk about some of the extra signals that are needed to get there. Throughout the endomembrane system, the proteins, after they're made, are actually passed from organelle to organelle, or to organelle and the plasma membrane via vesicles. And I think it's important to note that some proteins can also return. So if a protein is an ER protein, if it functions in the ER, it can travel to the Golgi, but be returned in vesicles to the ER. So these are the organelles that are linked together in this endomembrane system. So let's talk about how proteins end up in the ER. Okay, so recall with protein localization signal, we always have a zip code, we have a mail carrier, we have energy, and we have a mail slot. Okay, so this is true for proteins that are brought in to the ER. The zip code for the ER is an ER signal sequence. And this is a stretch of about 20 amino acids, and it's usually towards the start of a protein. So as I talk about this, I actually strongly recommend that you draw this pathway out. But let's talk about it. Okay, so here's our ribosome, and our ribosome is bound to mRNA, and we're synthesizing protein. Okay, so this green part coming out here is our protein, and some of the amino acids will make up this signal sequence, and that's shown here in red balls. So the red balls plus the green part is all a newly forming polypeptide. So just a little bit of the protein has been made. Once this signal sequence is made, the signal sequence will bind to this protein called the signal recognition peptide, or SRP. The SRP is the mail carrier. It reads this zip code. And importantly, and we'll come back to why this is important in a second, SRP is bound to GTP. And when it's bound to GTP, it's going to interact with this signal sequence. So far we have a signal sequence being made, the ER signal sequence, and this is our zip code, and it's been picked up by our mail carrier, that's SRP. SRP then binds to this SRP receptor that's embedded into the ER membrane. So the SRP here is in green, and the SRP receptor is in this blue color. The SRP receptor is also bound to GTP, and when they bind together, so the SRP and SRP receptor, this causes the hydrolysis of GTP, and it transfers this signal sequence to this pore protein called the translocon, okay? So now if you're following, our energy input was GTP hydrolysis. And if you look at this pore that's called the translocon, this is our mail slot. So this is how we're going to get, the protein is going to get through the membrane. So this whole process, right, when the SRP was bound to the signal sequence, and then it bound to the SRP receptor, the SRP was also bound to the ribosome, okay? And so it brought the ribosome with it. And so then the ribosome can keep making protein, but as it makes the protein, it feeds it through the translocon. Then once it's done, it releases the protein, and the mRNA and ribosomes dissociate, and the translocon closes. The final step in the ER is that the signal sequence is cleaved off, and then the protein folds. And so now we have a folded protein that's within the ER lumen. So here again is our protein being made at the surface of the rough endoplasmic reticulum. So this is what all those ribosomes on the ER, this is what they're doing. They're binding to SRP, and then SRP is transferring the signal sequence to the translocon, and then the protein is being made and folded. Once that protein is folded, it's going to be carried in vesicles to the Golgi apparatus, and it's going to enter into the cis Golgi. This is the location of the Golgi that's closest to the nucleus. And then it's going to mature in the Golgi and gain extra modifications like added sugars, and it's going to pass through the different layers of the Golgi, and it's going to exit at the transface. And as it moves, it's going to move through vesicles on the outside, and it's going to exit at the trans Golgi. And there's a few different fates for this vesicle as it leaves the trans Golgi. It can be embedded into the plasma membrane, and this is common if this is an amphipathic protein with a stretch, and it's actually embedded here already, and it just will get stuck in the membrane. If it's a luminal protein, like the one shown in this diagram here, this vesicle will just fuse with the plasma membrane and release the secreted protein. As I mentioned to you earlier, some proteins can actually get recycled back to the Golgi, or retained in the Golgi, or get recycled back to the ER. So ER proteins, Golgi proteins, plasma membrane proteins, and secreted proteins are all made in this system. The other location I'm going to talk about is proteins that are brought to the lysosome. And so this is an example of when an extra signal is needed. So each protein that comes out of the Golgi but is destined for the lysosome has a specific molecular tag. And so proteins that end up in the lysosome get modified in this Golgi with this special sugar residue called mannose-6-phosphate, or M6P. And proteins with this sugar residue are shuttled into these vesicles that deliver their contents to the lysosome. And so here we have our protein with our tag in the lysosome. And these examples of these proteins include about 40 acidic hydrolases. And so as we talked about in the parts of the cell, the lysosome has a really low internal pH because the lysosome has a proton pump in its membrane that pumps protons into the lumen and maintains the pH of the lysosome around 5. And this activates these acidic hydrolases, and this helps the lysosome recycle cellular waste. But how does cellular waste reach the lysosome? So materials can be brought into cells in these two different processes in which the plasma membrane is pinched off. Those processes are receptor-mediated endocytosis and phagocytosis. There's a third process in which damaged organelles are surrounded by membranes, and then those damaged organelles are brought to the lysosome for degradation, and that process is called autophagy. And I'm going to talk about each of these three processes. The first process I'll talk about is endocytosis, specifically receptor-mediated endocytosis. And in this example, a ligand, which is a small molecule, binds to a receptor. And when this happens, this triggers this inward budding of the plasma membrane. And when this pinches off, when this vesicle or this bud pinches off at the plasma membrane, this forms an endosome or an endocytic vesicle. And the cell can separate contents that it wants to traffic to the lysosome, or it wants to traffic back to the plasma membrane. And so sometimes some of the receptors are actually recycled back to the plasma membrane. But some of the contents will eventually mature into this late endosome with a lower pH and then fuse with the lysosome. Phagocytosis is a process in which larger material is brought into cells. And so this process is showing this inward budding of the plasma membrane. And so that's one way phagocytosis can occur. But we also see the plasma membrane stretching out and surrounding a target particle. So there's multiple ways that this can occur. But we get eventually this formation of this new organelle called the phagosome. And this contains the large ingested material. And this will be trafficked eventually to the lysosome. And this lysosome with the acidic hydrolases will break down whatever the cell has eaten. In this case, it might be a bacteria. And this can eliminate that bacteria as a threat and potentially free up some monomers to be used by the cell. The last pathway I'll talk about is autophagy. And so this is a pathway discovered not too long ago where it's a process where it's a repair process. So you can have a damaged organelle inside your cell, and this damaged organelle sends out cues to the cell, and those cues cause a membrane to form around the cell. Form around the damaged organelle. When the damaged organelle is surrounded completely, we call this the autophagosome. And this can also be delivered to the lysosome for degradation. So what I've told you so far is proteins destined for the ER, Golgi, lysosome, plasma membrane, or extracellular space are made in the ER. ER import, like nuclear import, requires a zip code, a mail carrier, energy, and a mail slot. Our zip code was the ER signal sequence. Our mail carrier was SRP. The energy came from the interaction between SRP and the SRP receptor, and our mail slot was the translocon. Proteins may have additional zip codes, like M6P, to reach the lysosome, and material is also trafficked into the cells and to the lysosome via endocytosis and phagocytosis. So that's it that I have for this video, and I look forward to seeing everyone in class. Thanks, bye.